Hello everyone. So today we are going to talk about the metabolism of the major lipoproteins that we have studied so far. So just going back one slide, we learned that we have chylomicrons, VLDL, LDL, and HDL. These are the major ones. So we are going to go and look at one by one and understand how they work and what they do in terms of delivering or managing lipid metabolism inside our bodies. So we're going to start with metabolism of chylomicrons, which, as we know, delivers dietary triglycerides to peripheral organs and tissues. So how does chylomicron actually do that? So we start with production of chylomicrons. As you can see in this slide, we are going to build up the whole story. So approximately 8 to 10 hours after a fatty meal, all chylomicrons are cleared from circulation. So, but how does that happen? So first of all, chylomicrons, they originate in the small intestine, obviously because they are formed once the dietary triglycerides are disassembled and absorbed by enterocytes. And we learned the chylomicrons, once they're formed in the enterocytes, they enter our body through the lymphatics. So once they enter the lymphatics and they reach the circulation, chylomicrons, at the beginning, when they are first formed, they contain quite a bit of triglycerides, as you can see here, TGs, and cholesterol. C stands for cholesterol, and there are other particles that are soluble in fat that also come from the diet, but mostly is triglycerides because it's the majority of the particle and some of the cholesterol. And we also learned that there is a apolipo or lipoprotein called B48 that is put on the chylomicron as a flag that will allow it to be guided where to go and how to be processed by your body. And also a little bit of ApoA. But this chylomicron, really, the first thing that it needs to do is interact in the circulation, in the blood, with HDL. And HDL, we know, that comes from liver. And liver actually constantly releases HDL in the circulation, which is a particle that contains a little bit of phospholipids and cholesterol, almost no or very little triglycerides, but it contains ApoE, ApoA, and ApoC2. So you can see here that once chylomicrons, the ones that are just formed, just arising from the lymphatics into the blood, they don't have ApoC2, they have very little ApoA. So what happens is that once they interact in the circulation, they collide or they get in contact with HDL in the circulation, they exchange material. Actually, in the beginning, what happens is that chylomicrons, they receive ApoE, ApoC2, and a little bit more ApoA. So now chylomicrons become a particle that contains all these 4 apoproteins that you can see here. So what's the relevance of this? Well, the importance of this is that chylomicrons, they are supposed to deliver triglycerides or lipids from the diet into the cells. So in order for chylomicrons to do that, they need to be able to interact with a protein or an enzyme called LPL, or Lipoprotein Lipase. And this portion here of the slide in yellow says no hepatic tissues, meaning that everything except the liver. So this includes muscle, heart, and adipose tissue. So what happens is that these tissues, they produce LPL, or lipoprotein lipase, as the name says, is an enzyme that breaks down lipids from lipoproteins, in this case, chylomicrons. So, but in order for lipoprotein lipase to do its job, it needs to interact with the particle, in this case, chylomicrons. It can only do this because on the surface of the chylomicron, you have ApoC2, which came from HDL that actually donated to chylomicron when it first interacted in the circulation. So now, because it has ApoC2, it can interact with LPL. So these two interact, and actually it anchors the particle with LPL. LPL is an enzyme that is produced by all these tissues, but it's actually sent out of the cell into the lumen of the capillary that irrigates that area. And LPL is actually hanging on the surface of the capillary that is irrigating that tissue. So now once chylomicron passes by and ApoC2 interacts with LPL, it kind of anchors the particle, so it stops at that point and allows LPL now to be active and promote lipolysis or breakdown of triglycerides in chylomicrons, and it releases fatty acids and glycerol. Remember, triglycerides, they are made by one backbone of glycerol esterified with 3 fatty acids forming the triacylglycerol. Now what LPL does is it breaks it apart again, separates fatty acids from glycerol. And this is important because the triglyceride molecule is big, and it wouldn't cross the cell membranes of these tissues. So now with lipoprotein lipase, you break down into smaller molecules, particularly those fatty acids that were present in triglyceride now get into the cells, and the cells can actually use it for whatever purpose. If it's in muscle, it can be used to produce energy. The heart really is a muscle that uses quite a bit of fat and uses fatty acids to produce ATP. And the adipose tissue actually uses this fatty acids to store again and form triacylglycerol. So some of these fatty acids that came from a triglyceride molecule now will get into the adipocyte. And if it's in abundance, more than what the adipocytes or your body uses for its metabolism, it will actually store again as triglycerides. So it will re-esterify fatty acids on a backbone of glycerol and make it as a molecule that is stored as triglyceride again in the adipose tissue. But all these non-hepatic tissues can extract these fatty acids from the chylomicron because of the interaction of LPL with ApoC2. And glycerol is released into the circulation, and this can go to the liver, and liver can convert glycerol into glucose. So it's a process called gluconeogenesis. But the important aspect here is that chylomicrons will donate their content of triglycerides, and once that happens, they become what we call chylomicron remnants. So they are much smaller. So you can see here this slide showing the change in size. So which was a big, rich in triglyceride particle, now it's a much smaller and with a much reduced content of triglycerides because the fatty acids were donated to extra hepatic tissues. So these chylomicron remnants now are formed in the circulation, and what happens is that they again interact, or they collide and exchange material with HDL. And when they do that, they give back ApoC2 and ApoA. So they give back what they gained to begin with when they first entered the circulation. So they give it back. So now these chylomicron remnants contain a small amount of triglycerides, some of the cholesterol that came from the diet, and only 2 apoproteins, B48, which is the one that was originally there, and also ApoE that it gained from HDL. So what happens is that especially ApoE allows this particle to interact with a receptor at the liver, and also with HL, which is hepatic lipase, which actually will interact with this particle and disassemble it. So inside of the liver, this particle now will be converted into its small, is going to be broken down into its constituents. In this case, glycerol that came from this circulation, but also amino acids that came from the circulation, cholesterol that was in the particle, and fatty acids that were also components of triglycerides. So again, the liver takes up all this and keeps sending out HDL particles containing those recycled products of this chylomicron, and this process keeps happening as long as you have chylomicrons in the circulation, or every time after a meal, this process keeps happening. But the important thing is that if you eat something now, 8 to 10 hours later, all these triglyceride particles that were formed, especially if it's a fatty meal, because if there is no fat in the meal, you will form very little chylomicrons. But when you eat a fatty meal, you're going to produce quite a bit of these particles, but after 8 to 10 hours, they're going to be completely cleared from the circulation. So that's I would say, the lifespan or the cycle that chylomicrons have to go through in order to deliver their content to the tissues. So this is the story basically with chylomicrons. Now the second particle that I want to talk about is Vldl. So Vldl is to some extent similar to chylomicrons, but the difference is that they deliver endogenously produced or generated lipids, and they come from the liver instead of coming from the enterocytes in the duodenum. They are generated in the liver, and they send lipids to peripheral tissues. And the other important thing about Vldl is that they give rise to LDL. So it's an important particle because it has some important implications to metabolism of lipids and health in our organisms. So let's start again from the liver, because this is where VLDLs come from. And why would the liver produce VLDLs? Well, because either if you have a diet that is rich in sugar, carbohydrate, a lot of the sugar that is surplus, let's say the extra amount of carbohydrate that you ate, that your body does not use for energy or for any other purpose, your liver can convert into lipids. Now, because liver makes lipids, it needs to also send those lipids to peripheral tissues, to other organs and tissues. And it does so by producing VLDL. So VLDL is a particle that comes from liver, and it contains ApoE, ApoB100. Again, it's an analog of B48. The difference is that the B48 that came from the duodenum had a molecular weight of 48 kilodaltons. Now, the one that comes from liver has a molecular weight of 100 kilodaltons. That's why it's called B100. And it also has ApoE, as I mentioned, and also ApoC2. It's a particle rich also in triglycerides, not as much as chylomicrons, but it's a significant amount. The majority of its content is made up of triglycerides and cholesterol. So they both came from the liver, or this particle comes from the liver. And again, once it gets into the circulation, it will interact with HDL particles, or high-density lipoproteins, that contains the same thing that I spoke before, phospholipids, cholesterol, and also some of those lipoproteins, ApoE, A, and C2. So what it does is that once they collide, once they interact, HDL will donate more ApoC2 and more ApoE to VLDLs. So it just enhances the content of ApoE and ApoC2 in the VLDL particle. So similar to, again, triglycerides, VLDLs now containing ApoC2 will interact in non-hepatic tissues with LPL. Same thing, LPL will promote the breakdown of triglycerides. So fatty acids will enter the tissues, and now glycerol is released into the circulation the same way as we learned in chylomicrons. Glycerol can, in the circulation, goes to the liver can use it to make glucose in a process called gluconeogenesis. Now, what happens after this donation of triglycerides takes place? So at this point, VLDL interacts again with HDL and donates back, gives it back to ApoC2 back to HDL. So what happens is that it loses ApoC2. If that happens, now it cannot interact with LPL anymore, and it will remain as it is. And this particle contains B100, ApoE, and is called intermediary density lipoprotein. It's shrunk, it's smaller than the original VLDL because it lost some of its triglyceride content. It is still rich in cholesterol, and it forms this intermediary density lipoprotein, which will interact again with HDL and give back ApoE, so it will lose ApoE too. So with that, some of these particles that lost ApoE, that only have B100, Now, some of it can actually reach the liver and be metabolized in the liver, but this is just a fraction of the particles that actually are circulating them end up doing that. Most of the particles now become LDL, and LDL is a particle that contains only B100. So this is the LDL particle. And what's the thing, the big thing with LDL? What LDL does, there is a receptor for it called Ldl receptor that is abbreviated here as LR. So LDL particle, low density lipoprotein, will interact with this LDL receptors and will donate its cholesterol content to non-hepatic tissues. So in other words, cholesterol now, that is the main component of this particle, will be donated to non-hepatic tissues. So when I say non-hepatic tissues, again, it's because everything except liver. So one of the concerns that people have with that is commonly raised with LDL is that because it donates cholesterol, and it could be muscle, heart, could be your blood vessels. And this is why, the reason why they say that LDL is considered bad cholesterol. Why? Because it has its cholesterol content will be donated to tissues. It could happen that this is the case, for instance, of your arteries, and that could actually increase the formation or promote the formation of atherosclerotic plaque. And because it donates its cholesterol content to tissues, it may be considered, or it's often called, bad cholesterol. But keep in mind, cholesterol is an important molecule to all organs and tissues, so it exists for a reason and for a good reason. But the problem is that when you have too much or too many of these particles and it's constantly elevated, and you have other risk factors, it may contribute to the generation of atherosclerosis, of plaque, clog the arteries, because the cholesterol content will be donated to tissues, including the artery. And the other, once it does that, it can also be cleared from the circulation, and this is one of the cases, because it can also not only interact with the non-hepatic tissues, but can also interact with the liver, because liver also has LDL receptors. So once it gets to the liver disassembles this whole particle, takes the cholesterol and fatty acids, and uses it to make different molecules or different products. One of them could be, again, reassemble this whole thing and send Vldl out again, or could you use, for instance, cholesterol to make bile, or could you use fatty acids to make for oxidation, for instance, to produce energy for its own metabolism. So what differs between chylomicron and VLDL is that chylomicrons are completely cleared from the circulation, whereas VLDL gives rise to LDL, and LDL now can distribute or donate its cholesterol content to tissues that could, in some cases, be associated with the development of cardiovascular disease. And that's why it's normally called or named bad cholesterol. But keep in mind, it exists for a purpose. So the third particle we are going to talk now is HDL, or high density lipoprotein. And the main feature of HDL is what we call reverse cholesterol transport. And we're going to learn exactly why it's called reverse, and also why this particle is normally named as good cholesterol, because it does actually the opposite of what LDL does. You'll see, we are going to learn it next. So let's start this diagram again from the liver, because the liver is the main organ or main producer of HDL, or high density lipoprotein. So liver contains phospholipids, cholesterol esters, and it can use it to make bile. As we know, bile is an important product for digestion in the intestine. And the intestine also makes some HDL. It's a small portion. That's why we have a much thicker arrow pointing coming from liver than we have from the intestine. But intestine also makes some HDL. It's much smaller contribution than liver. But in the circulation, we have this HDL now that is just first created, or nascent. We call it nascent because it just arisen. And it's called discoidal. Discoidal because it's kind of flat. It has a small content of cholesterol. It has more phospholipids. It's rich in phospholipids and cholesterol, but in small amounts. So instead of being a circular or discoidal, spherical shape, it's more discoidal because it's flat. It's not loaded with lipids when it's first created from liver. So what happens is that, again, HDL travels in the circulation. Not only it interacts with other particles, as we learned before, but HDL on its own, travels in the circulation, and it goes to what we call, again, non-hepatic tissues. Adipose, muscle, could be the arterial wall, it could be an atheroma plaque. So all these different tissues can actually interact with HDL. And what happens is that HDL anchors to the cells in these tissues via ABCA2 or ATP binding domain, or it's a cassette that is a protein that is localized on the surface of these tissues and allows the HDL particle to anchor. And what it does is it collects cholesterol from the tissues. Instead of donating cholesterol, it actually takes it up, removes cholesterol from all these tissues. And that's why we call it a reverse cholesterol transport, because it actually takes it away from the tissues instead of donating to tissues. And obviously, because if you have a high number of HDL particles in the circulation, you have a high capacity to clear cholesterol from peripheral tissues. And for that reason, we consider HDL a good cholesterol, because it's rich in cholesterol, but it's removing it from the tissues. And for that reason, it's normally associated with a positive response in your body. And the way how HDL takes up lots of cholesterol is because it has this enzyme called LCAT, or lecithin cholesterol acyl transferase. So what it does is it takes cholesterol. As the concentration of cholesterol within the particle increases, it converts it into cholesterol esters. In other words, it takes some fatty acids, for instance, and esterifies with that cholesterol and makes a cholesterol ester, which is a way for this particle to reduce the concentration of free cholesterol. So this way you don't create, or you allow a gradient of cholesterol in which its higher concentration is higher in the tissues, and not as it doesn't impair or doesn't stop coming into the particle. Because if you build up lots of free cholesterol into the particle, it comes to a point that the gradient now, instead of being favorable to receiving cholesterol, is going to be favored to donating cholesterol. So in order for that not to happen, what happens is that the particle contains LCAT. LCAT converts cholesterol into cholesterol esters, shifts, again, reduces the gradient of free cholesterol in the particle, and then more cholesterol keeps going into the particle. So that's a very effective way to clear cholesterol from peripheral tissues. Now, what happens is that this particle becomes, instead of discoidal, becomes spherical. Why is it? It grows in size because now it becomes rich in cholesterol esters, free cholesterol, and the same phospholipids that were there before. But it gained all that from peripheral tissues, became a bigger particle, became more spherical in shape now, and it can travel to the liver. There is a receptor mediated interaction. So this particle interacts with the receptor, donates phospholipids, cholesterol, and cholesterol esters to the liver, and it detaches from the liver. It's not destroyed on the first passage, if you will, or interaction with the liver. It actually detaches once it unloads its cholesterol, phospholipid, and cholesterol ester content, it becomes again discoidal because it shrinks in size once again, detaches from the liver, and it goes again to collect more cholesterol in another. It keeps doing it several cycles of this before the actual HDL particle becomes or is degraded or destroyed by the liver. And this process is called Hdl cycle because it keeps going on, taking cholesterol from peripheral tissues, giving it to the liver or delivering it to the liver, and then recycles back to go and do that more. So this is called HDL cycle. So as you can see here, there is a good reason for people to call this a good cholesterol particle, because it really clears up cholesterol, removes cholesterol from peripheral tissues, take it to the liver. Liver can use it to make bile acids, for instance, and send this cholesterol back into the intestine for digestion, and at the same time clears cholesterol or reduces the amount of cholesterol that is available to peripheral tissues. So this is a cycle that keeps happening, and for that reason it's called good cholesterol. Now, we mentioned here that HDL interacts with the particles, and it gives, at some point, it gives apoproteins, ApoC2, ApoE, and ApoA to chylomicrons and Vldl. But when it does that, not only, I spoke about half of or half of what it does, which is donate lipoproteins to this other particles, but actually there is an exchange of material. For instance, HDL, when it interacts with chylomicrons, it takes phospholipids, and this is what phospholipid transfer protein. So in other words, it gives ApoC2 and ApoE and ApoA to chylomicrons, but it also takes from the chylomicrons phospholipids, and this is because there is an enzyme called phospholipid transfer protein that transfer phospholipids from the chylomicrons to HDL. And HDL transfers in exchange cholesterol esters to chylomicron. Cholesterol esters are those cholesterol molecules that were esterified, for instance, with fatty acids that it collected from peripheral tissues. So it gives it back to chylomicrons. So chylomicrons can actually use this cholesterol ester to donate to peripheral tissues, but it can also, because it's cleared by the liver will get rid of it once the cycle of chylomicron is complete. So there is an exchange not only of particles, but also an exchange of lipid content between these two. And a similar thing happens with Vldl. Vldl receives cholesterol esters because of CETP, cholesterol ester transfer protein, and Vldl gives phospholipids to HDL through or via the activity of phospholipid transfer protein. And these proteins, they are in the circulation, so once there is an interaction between these particles, these proteins can then act and facilitate this exchange between HDL and chylomicron and Vldl. So this is an important also aspect of the exchange and how lipids are actually managed into the circulation. And the last mechanism I want to talk about is what we call proteins mediated by a protein called proprotein convertase subticillin caxin 9, for short PCSK9. I'm not going to ask you to memorize this big name. All you need to do is abbreviation PCSK9. It's a secreted protein. So again, it's produced by the liver, and it binds the LDL receptor on the surface of hepatocytes. Remember, hepatocytes have LDL receptors. When the LDL binds to it, it internalizes the whole particle, and the LDL is internalized in the liver, in the hepatocyte, and it's destroyed in there. So all the content of that LDL particle will be released inside of the liver. And this leads to intracellular degradation of the LDL receptor in the lysosomes of hepatocytes. The whole thing, it's the LDL particle, the LDL receptor, they bind. Once they bind, they're internalized, and both of them are degraded. Now, when you have this PCSK9, what it does, it binds the LDL receptor, and the LDL receptor now is internalized without binding to any LDL particle. You remove actually the LDL receptor from the surface of hepatocytes. So this serves as a mechanism for the liver to regulate how much cholesterol it will take in. So if there is lots of cholesterol already in the liver, PCSK9 is released, it binds to the receptor and prevents LDL particles from binding to their receptor and donate their triglyceride or their cholesterol content to hepatocytes. That's because the assumption here is that the hepatocyte already has plenty of cholesterol, so in order for not to take more, it sends a signal outside that we will actually suppress the binding of LDL particles with the receptors, and therefore it will degrade the receptor and not allow the transfer or the clearance of LDL particles by the liver. So PCSK9 is believed to be a mechanism by which the liver regulates how much cholesterol it will take in or not. So what the pharmaceutical industry did is like, well, if we can inhibit PCSK9, we can prevent the liver from not clearing LDL from the circulation. So we could actually reduce LDL levels or the bad cholesterol from the circulation if we inhibit PCSK9. So that's a novel therapeutic mechanism that has been developed with the idea of lowering LDL cholesterol from the circulation. So I have a diagram here that kind of, let's see if we can explain it better. So again, this is a liver cell. Okay, so let's look here. You have cell membrane that is in this space in between these two black lines. So it's a gray area, which is the intermembrane space here. So we have this liver cell. So what happens is in the blood, you have in yellow here, we have all these LDL particles. And on the surface of the cell membrane, you have LDL receptors in red. So what happens is that LDL binds to its receptor. It's internalized in the cell. So and what happens is that this vesicle contains both the LDL receptor in red and the LDL particle. So now what happens is that the LDL receptor is released from this complex, goes back to the membrane, and recycles itself. So it keeps doing this, keeps binding to LDL, and LDL is internalized. So what happens is that once the LDL separates from the LDL receptor inside of the cell, the LDL now is degraded by the lysosomes. Lysosomes are organelles in the cells that contain lots of digestive enzymes. So what they do is they take everything apart. So they break down that LDL, and the LDL is disassembled, and with this you clear it from the circulation. So, but the receptor keeps going back and being recycled. Now, but let's say that the liver cell, the hepatocyte, says, oh, we have plenty of cholesterol now, we don't need more. It's full of cholesterol. I want to stop taking in cholesterol. So what it does, it releases PCSK9. You see here, it's produced by the endoplasmic reticulum in the cell. So PCSK9 now is in vesicles, it's put in vesicles, and the hepatocyte releases PCSK9 in the circulation. So in pink, you see, or magenta, you see here the PCSK9 now in the circulation. So what it does, it goes and it binds the LDL receptor, the same LDL receptor that you have here. So now you're binding them on the surface of the cell. So when it does that, it prevents LDL from actually binding. Because it does so, now LDL stays in the circulation. So now what happens is that the binding of PCSK9 with LDL receptor forms a complex. It goes again, is internalized. Now, instead of the receptor being released to recycle, to go back to the membrane, now the whole complex goes into the lysosome. So what the lysosome does is it digests everything, so reduces the number of receptors that are available for LDL to circulate, or sorry, of LDL, for LDL to bind and to be cleared from the circulation by the hepatocyte. So the liver kind of uses this as a mechanism to regulate how much cholesterol it will take. And again, the idea of the pharmaceutical industry is to come up with a medication. In this case, is actually an antibody. It's called monoclonal antibody that actually binds to PCSK9. So by binding here, it doesn't allow it to bind to the LDL receptor. So the LDL receptor stays on the membrane and keeps recycling LDL. So that is used as a strategy to reduce or to lower LDL from the circulation. So that's the idea or the principle behind the use of this medication or this pharmacological agent to try to stop or prevent the liver from saying, I don't want to have more cholesterol. So it keeps sending cholesterol to the liver, and the liver will have to deal with it in different ways. But this is the idea behind it. So again, PCSK9 and the pharmacological agent that has been developed is a monoclonal antibody that binds to it in the circulation and prevents it from binding to the LDL receptor. Okay, so that's the last, I would say, mechanism that regulates lipids in our organism. And to finalize this, what we have is the normal values and ratios of circulating lipids. Again, there is no need for anyone to memorize these numbers. I just have this table here because it gives you a reference. What are the normal values? For instance, what is normal for triglycerides to be in a healthy person? So it's between maximum, let's say maximum of 210 milligrams per deciliter. Cholesterol is between 50 and 200 milligrams per deciliter. HDL, the so-called good cholesterol, so it's anything between 30 and 90. Obviously, the higher we tend to attribute or say that it's the higher the better. So I've seen some people with values that are quite high, but some on average is between 40, 45 is what you normally see. You have VLDL, it's about 5 to 40 milligrams per deciliter, LDL 50 to 140, and there is a ratio of cholesterol HDL. So what you do here is you not only look at the absolute values, but you look at the ratio, because if you have a high level of cholesterol, high amount of cholesterol, but you also have high amounts of HDL, it means the HDL is actually clearing cholesterol from circulation, so it's good. So if you have a low ratio, it means that you have cholesterol, but your HDL is high. So if this ratio is low, it's good. So here it's about 3.7 to 6.7, which is usually the ideal or the optimum ratio that we look for. So if the ratio is too high, it means that your HDL is low, but your cholesterol is high. So which means that most of the cholesterol you have is in the LDL particle, not in the HDL particle. So for that reason, the ratio can also be used, and is often used as a means to interpret what's going on with lipid metabolism. And you also have the LDL-HDL ratio. So you also want to have a low ratio here, because in this particular case, if the ratio is low, it means that you have low LDL and high HDL. So most of the cholesterol or the transport is in the HDL and not as much in LDL. So the normal range is 3.3 to 4.4. So if it's much more than that, it's probably because you have low HDL and high LDL. In other words, you have low amounts of what's called bad cholesterol, low amounts of what's called good cholesterol, and high amounts of the bad LDL cholesterol. So again, no need to memorize this. All I want, if I'm going to ask you questions about this in the exam, I'll give you the numbers. What I want you to understand is the importance of what it means and how you use this. Don't worry, you don't need to memorize numbers. I always, if I ask questions involving numbers, I'll give you the numbers and ask you to interpret that. So no reason to memorize them. Even the ratios, I'm not going to ask you to memorize the ratios. What I want you to understand is what the ratios mean If I tell you what the ratios are, then I want you to tell me what's going on, or probably have an idea of what's going on there. So with that, we are gonna stop here. And this is the last slide that we discuss about lipids. So this ends the lipid section of this course. And the next 4 lectures, if I'm not wrong, will be taught by Dr. Joss, and it will be about protein. So I'll be back after the protein section is finished. But for now, thanks for watching, and I'll see you soon. Bye now.